Hello students, welcome to readmedprep Academy channel. Today we are going to discuss in genetics principles of inheritance and variation part 1. In this we are going to talk about Rh blood groups, Rh factor. What is Rh factor? Rh factor or Rh antigen is found on the surface of erythrocytes. It was discovered in 1940 by Karl Landsteiner and Alexander Wiener in the blood of rhesus monkey, the macaca rhesus, and later in the human beings. Who is Rh positive? The term Rh factor refers to the immunogenic D antigen of the Rh blood group system. An individual having a D antigen or Rh D positive, designated as Rh to the power of plus, and those without D antigen are Rh D negative, designated as Rh to the power of minus. We can see that the Rh factor antigens present on the surface of the RBCs, and the Rh negative blood will not have any antigens on the surface. Now, when we take O negative blood, there is no A antigen or B antigen or Rh antigen. When you take A negative blood, the surface will contain only A antigens and there are no Rh or D antigens on the surface. When you take a B negative blood, the surface of the RBCs will contain only B antigens. There are no D antigens or Rh antigens. When you take AB negative blood group, the red cell surface antigens are A and B. There are no D or Rh antigen. When you take O positive, the surface of the RBCs do not contain A or B antigens, but they have Rh antigens on the surface. When you take A positive blood, the surface of the RBCs will contain A antigens as well as R antigens. When you take B positive blood, they have B antigens on the surface of the RBCs in the membrane and the Rh antigens. Whereas when you take the AB positive, the A antigens are present, the B antigens are present. In addition, there will be D antigens or Rh antigens on the surface. How this Rh factor is inherited? Rh factor in the blood is inherited as a dominant trait. Naturally occurring anti-D antibodies are absent in the plasma of any normal individual. Now you see this diagram. We have the father who is homozygous for Rh positive, which is represented by capital RR, and the homozygous Rh negative mother, which is represented as small r. The father will produce the capital R gametes, and the mother will produce ovum which contains small r. The progeny that is formed is 100% Rh positive with capital R and small r, since capital R is a dominant allele. If you take the parents as a homozygous Rh negative mother and a heterozygous Rh positive father, the homozygous Rh negative mother is represented as small r and small r, and they produce gametes small r and small r. Whereas the heterozygous Rh positive father, which has a capital R and a small r, will produce sperms like capital R and small r. And the children that are formed are capital R and small r and small r. That means 50% of the population will be Rh positive and 50% of the population will be Rh negative if the homozygous Rh negative mother marries a heterozygous Rh positive father. When a heterozygous Rh positive father marries a heterozygous Rh positive mother, the heterozygous Rh positive father will produce sperms containing capital R and small r, whereas the heterozygous Rh positive mother will produce capital and small r ovum. And the progeny that is formed is 25% Rh positive, 50% Rh positive and 25% Rh negative. That is 75% of the population will be Rh positive and 25% of the population will be Rh negative. Out of the 75% of the population, 25% of the population will be homozygous and 50% of the population will be heterozygous. This is the inheritance of the Rh antigen. When anti-D antibodies are formed in the body, if an Rh negative person is exposed to Rh positive blood cells or erythrocytes for the first time, anti-D antibodies are formed in the blood of that individual. For example, if a Rh negative mother bears a fetus of Rh positive child, the first pregnancy, the mother is exposed to the Rh factor and prepares or makes antibodies to the Rh factor. On the other hand, when an Rh positive person receives Rh negative blood, no effect is seen. What is the genetic control of the Rh factor? That is explained by various hypotheses. Let us discuss about the Fisher and Race hypothesis. Rh factor involves three different pairs of alleles located on three different closely linked loci on the chromosome pair. This system is more commonly in use today and use the CDE nomenclature. What are the Rh alleles mentioned in this hypothesis? In Fisher and Race hypothesis, Rh blood types, three pairs of Rh alleles are used. Capital C small c, capital D small d, capital E small e, which occur in three different loci on the homologous chromosome pair. What are the Rh genotypes? The possible genotypes will be one capital C or small c, one capital D or small d, one capital E or small e for each chromosome. For example, capital C, capital D, capital E with small c, small d, small e. Then next is capital C, small d, capital E with small c, capital D, small e. Next is small c, small d, small e, small c, small d, small e. Now the fourth you can see capital C, lowercase d, lowercase e with lowercase c, lowercase d and capital E, etc. What are the Rh phenotypes according to this hypothesis? Fisher and Race hypothesis have mentioned that capital D allele is the dominant allele and will produce Rh positive phenotype. And the double recessive genotype small d will give rise to Rh negative phenotype. So to sum up the Fisher-Race hypothesis, Rh inheritance is controlled by three closely linked loci on each chromosome of a homologous pair. Each locus has its own set of alleles which are capital D small d, capital C small c and capital E small e. The D gene is the dominant gene to the small d gene, but capital C small c, capital E small e are codominant respectively. The three loci are so closely linked that crossing over does not occur, and the three genes on one chromosome are always inherited together. Now let us move on to the another hypothesis called Wiener hypothesis. Wiener proposed the existence of 8 alleles. Capital R to the power of 1, capital R to the power of 2, capital R to the power of 0, capital R to the power of Z. Small r to the power of 1, small r to the power of 11, small r to the power of y at a single Rh locus. So according to the Wiener's hypothesis, the genotypes and phenotypes are all genotypes carrying a dominant gene, capital R allele. Capital R to the power of 1, capital R to the power of 2, capital R to the power of 0, capital R to the power of Z will produce Rh positive phenotypes. Double recessive genotypes like lowercase rr, r to the power of 1, r to the power of 1, r to the power of 11, r to the power of 11, r to the power of y, r to the power of y, all being the lower case will give rise to Rh negative phenotype. Now, comparing the Wiener hypothesis and the Fisher-Race hypothesis, what are the antigens present? R to the power of 0 will produce capital D, small c, small e. The antigens present are capital D, small c, small e antigens. R to the power of 1, capital D, capital C and small e. The antigens present are capital D, capital C and small e. R to the power of 2, capital D, small c, capital E, and the antigens present are capital D, small c and capital E. R to the power of Z, which is the capital alphabets of D, C, E. The antigens produced are capital D, capital C, capital E. Now, the recessive allele R, the Fisher-Race DCE is the formation of alleles and the antigens present are CE. R to the power of 1, capital C with small d and E, and the antigens formed are capital C and small e. R to the power of 11, the small case D, C and the alphabet E is capital, and the antigens produced are small c and capital E. R to the power of Y, when we take according to the Fisher-Race, it is small d with capital C, capital E. The antigens are capital C and capital E. Now, what is the incompatibility of Rh factor which causes erythroblastosis fetalis? So, Rh incompatibility has a great significance in childbirth. If a woman is Rh negative and the man is Rh positive, the fetus produced may be Rh positive, having inherited the factor from its father. You can see here that the Rh positive fetus is present and the mother is Rh negative. What happens? Rh sensitization occurs. When the Rh negative mother becomes sensitized by carrying Rh positive fetus within her body. How? By the damage of the blood vessels during childbirth, or because of the mother's immune system that recognizes the Rh antigens and gets sensitized. Sensitized mother produces Rh antibodies. The antibodies are IgG type, which are small and can cross the placenta and enter the fetal circulation. But by the time the mother develops the antibodies, the child is delivered. So the antibodies to the anti-D remains in the plasma. For example, you can see in this picture that the red blood cells that contains the Rh positive antigens enter into the mother's circulation and the mother is Rh negative, and these antigens cause Rh antibodies to form in the mother. So usually there is no effects associated with the exposure of the mother to Rh positive antigen during the first childbirth. What happens in subsequent pregnancies? Subsequent pregnancies, when the child is Rh positive, carried by the same Rh negative mother, the child is exposed to the antibodies that had been already formed in the previous pregnancy against the Rh antigen. And these antibodies are carried across the placenta to the fetal circulation. These antibodies react with the antigens that are present on the surface of the fetal RBCs, resulting in the causation of hemolytic jaundice and anemia. This condition is known as erythroblastosis fetalis or hemolytic disease of the newborn. You can see in this picture that the Rh positive father and Rh negative mother during pregnancy, the first pregnancy causes the antibodies to be formed in the mother, and these antibodies remain within the mother. In the subsequent pregnancies, these antibodies attack the Rh positive fetal RBCs and causes the erythroblastosis fetalis. The antibodies made by the mother can cross the placenta and cause breakdown of the red cells in the fetus. This can result in hemolytic disease of the newborn with anemia and jaundice, which can be mild or severe. Very severe hemolytic disease of the newborn can cause hydrops and fetal death in the womb, or brain damage after birth from Kern icterus due to the very high levels of bilirubin that is formed in the blood. So how this disease occurs? The critical amount that is required is 0.1 milligram of the antibodies. The Rh antibodies that enter the fetal circulation coat the RBCs and they cause destruction of the fetal cells of the fetus by fetal reticular endothelial system, and this causes fetal anemia. Then there is fetal hypoxia and stimulation of the erythropoietin. Extramedullary lead cells synthesis occurs, and there is hepatosplenomegaly. There is hepatic cell failure resulting in hypoproteinemia, increased intrahepatic pressure and portal hypertension. Because of this, the fetus may have ascites, edema, hypoxia, placental thickness, polyhydramnios and pericardial effusion. How to prevent erythroblastosis fetalis? If the mother is Rh negative and the fetus is Rh positive, anti-D antibodies should be administered to the mother at 28th and 34th week of gestation as a prophylactic measure to prevent this disease. So if the Rh negative mother delivers a Rh positive child, then anti-D antibody should be administered immediately to the mother soon after delivery. What is the dosage? 300 micrograms should be given of the anti-D immunoglobulin IM or IV. What type of immunity develops? It's a passive immunity and prevents the formation of anti-D antibodies in the mother's blood by destroying the Rh fetal RBC before the mother's immune system is sensitized. This has to be done whenever the mother becomes pregnant. So today we discussed in genetics the principles of inheritance and variation part 1, Rh factor and genetic control and Rh incompatibility. Thank you very much. Kindly subscribe, like, share and comment the channel Read MedPrep Academy. Log on to readmedprepacademy.com. Our Facebook ID Read MedPrep Academy. Email is readmedprepacademy@gmail.com. Kindly post your questions in the comment box. We will reply with appropriate answers. Thank you.